With that, I'd also now like to welcome Brad Margus. He's going to speak with us this morning on the 10 things that you need to do to pursue research. Brad is a member of the Global Genes Board and is also the founder of the AT Children's Project. Please help me welcome Brad Margus.
Hi. So it's really an honor to be here, and I'm really humbled because I know some of the people in this room from many other things, and many of you have done much more than I, so it's kind of nuts that I'm up here and somehow supposed to be an expert teaching things. Pat Furlong, I was trying to think what word would I use to describe her, and I thought admirable and amazing, but then I thought what my sons would say, and the best word would be beast. She's a beast, okay? So what Pat's gotten done is absolutely incredible. So the first thing I'd tell you is I'm not a scientist, and I don't pretend to be, and you'll soon figure out that I shouldn't be here at all. But a lot of people in this room maybe sometimes feel that way. I wasn't a Silicon Valley success story like Matt, and I wasn't a nurse who took the MCATs and had a husband who was a physician like Pat. I'm just the guy. So what I'm going to do is tell you my story, and then for the first half, and then after that we can have some discussion and maybe or questions. So I went to Harvard Business School. Most of my classmates went to, and sorry, by the way, those of you in the room who have heard my story before, a few of you I know really well, please don't throw up. I know it's nauseous to hear it again, but this is my life, okay? And you'll see that I weave in a lot of different things about my life, my career, and my volunteer stuff, but you know how it is. It all gets mixed together. So on that note, I went to Harvard Business School. All my classmates went to Wall Street or investment banking or consulting or something, but I did something else. I did what was called a leveraged buyout, where you borrow money from other people and you buy a company, and it was a shrimp processing company. And you shouldn't be laughing. Anyway, now that you've all had a time to ponder it, I want everybody to say what you're thinking. Brad Gellin, right? So that's me. And hopefully I have an IQ a little bit higher than he did, but you guys can decide at the end. Anyway, I had this shrimp company. It was actually very profitable. Very quickly, I married a very beautiful woman and had 3 sons in 3.5 years. Everything was going really well, except for the fact that being Forrest Gump was a little embarrassing. My career was doing really well. And no detours were expected. And then, like many people in this room, we got a diagnosis. In this case, our middle son, Jarrett, in the middle, and Quinn over there on the right, were diagnosed with a disease. It's a brutal disease we had never heard of. It's called Ataxia. Ataxia. It's really rare, about 400 kids in the United States with it, and we were told it was an orphan disease. It affects a lot of different systems in the body. The part of the brain, the cerebellum, deteriorates so that you lose muscle control. About 80% of kids have immune problems, cancer, lots of other stuff that goes on. Here's some pictures of a healthy kid, 12 years old, and a kid with AT. You can see that spot called the cerebellum is much smaller and shrinking. Kids are totally normal when they're little. They run around. Around age 2, they become a little wobbly. They end up continuing to deteriorate, often misdiagnosed for cerebral or for ataxic cerebral palsy, but sooner or later the deterioration becomes obvious and people figure out it's genetic and hopefully diagnose it correctly. You lose control of your eyes, swallowing gets difficult, speech and it starts to go, and some kids develop tremors. All the while, cognitively, they're fine, so my sons can still beat most people in chess, invest in Ameritrade, or beat me in fantasy football. But physically, they're completely debilitated. It's very frustrating for them. Immune problems, the kids have a lot of the immune system problems that you have in other immune deficiencies. And about 30% of kids also, on top of that, have develop cancer. If they get it when they're young, it's usually lymphomas, leukemias, blood cancers. The older kids get solid tumors. And if the kids don't die from cancer, the most common causes of death are lung problems, where the bellows function gets so weak, and immune deficiency causes pretty mild infections to turn into pretty bad pneumonias. So kids die pretty early. So it's a pretty bad diagnosis. I know that usually I can get donors to suddenly really feel sorry for me at this point, but I know in this room, all of you have lots of problems, and you all have your own stories. So I kind of feel more at home today. So as I said, it's a really rare disease, about 400 kids in the US. They're turning over all the time. So I wanted to do something when we were diagnosed. There was no global genes yet. I secretly put Nicole Boyce's picture in my slide deck so she wouldn't mess with my slide deck. She sent me a note saying she loved my slides. I don't think she actually looked at them. So what did I do? And here's what we're all here for, is to learn what each other does in these situations. So it's a short list. I learned the science, learned not just the science. I had a couple PhDs tutor me very deeply but narrowly on just the science I cared about, which in our case was to start by finding the gene that causes disease. It's really different when you're learning because you need to. But I think equally as hard was to learn how the science works. I didn't know anything about how the government funds biomedical research. I didn't really care. I didn't know about the careers scientists have and how publishing is important, or how you have to get a postdoc and serve your time as a lab rat before you someday become a big shot. I didn't know any of that stuff. I didn't know who the good scientists were, the bad ones were, how to tell a difference. I didn't know what turned them on, whether they wanted glory, money, whether they wanted to, whether you could get to them by showing them a picture of a cute little kid who's dying, or whether they were nerds that really wanted to solve a puzzle, and if you could appeal to that, you could get them. But I studied them, and I also had to learn how science is funded and how to raise money. And that last part, by the way, I was a businessman. I knew how to talk to investors. I never was the type to go raise money as putting my hat out and begging for stuff. And my wife and I certainly weren't like an affluent couple who needed a cause or something. We were really busy with our kids, and we didn't envision being fundraisers. But nevertheless, like many people in this room, I went ahead and started a nonprofit organization, and we called it the AT Children's Project, and the mission was just to somehow improve the lives of kids with AT. So I'll come back to that mission because it's important to remember what your mission is. Sometimes it strays a little bit. So again, imagine trying to raise money for a disease that has only 400 kids in the US. As far as we know to this day, and I have to tell you just in this room that I still fantasize about a celebrity or a really, really rich billionaire having a kid with the disease. It's terrible, but I do. But so far, nobody like that has come along. And so it's hard, and ataxia telangiectasia is not a catchy disease name. Nevertheless, we started having fundraising events. We talked mostly just going after other families with kids, and then people who were, and as many of you know, when you have a rare disease, you have to appeal to people who have no personal connection with the disease. So we talked to anybody we could. If someone wanted to raise money for us in a garage sale that might raise $50, we told them to do it. We never told people no. What usually happened was those people would have a garage sale and raise $50, but someone would come to that garage sale and say, you really stink at this. You're pathetic, but I know how to run these things to raise money. Let me do it for you next year. And the next year they would raise, 10,000 or something. This list is just a short list. We have a lot of weird, bizarre things people have done, but it's all grassroots. We've had the usual running stuff. I've actually run a marathon for AT. Just so you know, before you all laugh, I usually lose about 30 pounds before I do that. But I have learned that compared to some of these hard-body guys that you see up there, a middle-aged fat guy can raise a lot more money. In fact, at the last time I ran, I had people offering to sponsor me if I didn't run. We had this guy, Tim Borland, who actually is from pretty close to here. He ran 63 marathons in 63 consecutive days. And we got a lot of press for it, and we scheduled kind of fundraising events all along the way. The guy was an amazing, amazing athlete. He traveled in the RV that you see there that we had him go across the country in with his wife and his 2 little kids. And when the whole 63 days was over, the last one he ran the New York Marathon. And when the whole thing was over, he informed us that his wife was now pregnant. So can you imagine that? Figure that out. What an athlete. We had the guy named Hunter Kemper. Hunter Kemper's a 4-time Olympian triathlete, and he's kind of adopted our cause. We got a nice thing on the side of the Wheaties box when he was on it that helped us out some. I'm just giving you some examples of different things we've done. We've had runs all around the country. We never imagined our logo showing up in some bizarre places like on motorcycles and motocross. But a family in Georgia the little girl had a thing to do that. A guy in France decided to race in his usual race across the Atlantic, but he did it for AT to raise awareness. In each of these cases, I'm forced to learn about something I know nothing about, and it's really awkward. There's a horse show that's held every year for us in Palm Beach, Florida, with very, very affluent people and fancy, fancy things, and I knew nothing about horse shows, so once again, I had to talk to the horse people beforehand and say, can you just give me 3 sentences I can say so when someone comes up with their beautiful horse, I don't just say, nice horsey, but I can actually say, that's a cool whatever, or that's a good thing. And I've now done that with horses and sailing and several other things. But you do what you have to do. Recently there was this event, a bowling event, and I just thought I'd show you the cool cookies that they made. On the left is another walkathon in Canada, and the middle or on the top right is a meatball smackdown, and the middle one there is one I really, really love because it's a doughboy dash where you actually run like 100 yards and then you eat a big like donut-like thing, and then you run another 100 yards, and that's my kind of race. We've increased awareness, and I think later when we're having a discussion we can talk about when this is useful and when it really isn't, but we got a lot of press early on. Barbara Walters did a 1-hour special on my family where camera crews followed us for like 18 months, and my 4th son was born on TV and everything. Great, like 13 million people watched it. We didn't get people sending in, tons and tons of checks from that, and that's what we had fantasized. We did hear from about 400 people who thought their kids might have AT, and about close to 50 did, so that was really valuable to us. But there's I think an art to how you use publicity, and sometimes it's kind of a knee-jerk thing that every board of every nonprofit says, we need to have more PR, and you really have to think, how is it going to benefit you? This is a cool thing up in Maine, a family that works for Walmart, got Walmart to do a thing that gave us a lot of awareness. And we've also gotten some famous people to help us along the way, just through serendipity and everything. Again, it's one thing to have a celebrity that might come to your event, and it's wonderful if they do because they make all of our other donors there feel good, and a great thing for them. But if they can't promise you that they're going to be at that event, like because of their filming schedule, they can only say they might be there, you lose all the big bang because you can't sell tickets to come see Matt Damon or someone like that. So there's it's a challenge using celebrities, but we've done what we could. Just so you know, Jennifer Garner is being really chummy with me in this picture because someone offered to donate $1,000 if I had a picture of me with her like that. So, if anybody else wants to make that donation, I'd like to call Jennifer right now. So, Brad Paisley, who's a Grammy Award-winning country western singer, actually recorded a song that the lyrics were written by a kid with my son's disease, Joe Kindergan. That kid died last month. It's really sad. Anyway, now that I gave you an introduction to our organization, I'm going to tell you how we've tried to accelerate AT research. Again, I'm not trying to be the authority here, and my thing's going to be kind of focused on a neurological disease because that's what I think about the most. But maybe you can learn some things from it, or maybe it'll trigger some things that we can all discuss, or maybe you can come up with some better practices for some of these things I've done. But one thing I can say is that although I wasn't trained background-wise at all, I think sometimes that's helped because it turns out you can get a lot in the world just by being a needy person and saying to people, I really need your help. And hopefully you'll see that. So the first way we've orchestrated a lot of research, of course, is by giving grants that you've heard about. This is an example, the Yossi Shiloh lab in Tel Aviv, Israel. They cloned the gene for AT. Before that, we didn't know if it was 1 gene or possibly 4 genes. We figured out it was 1 gene. Getting samples was a really big thing in the beginning. It turned out that there were 2 researchers, one in California and one in New York, that were collecting lots of blood samples from families with AT kids to try to do what's called linkage analysis or positional cloning to find the gene. And it turns out that neither of them were really geneticists, but they could collaborate with geneticists by having these samples as their currency. And what really killed me when I first started doing this was that the two guys weren't even talking to each other. So we changed that right away. We set up a cell line bank at Coriell Institute, which is funded by the government, and quickly I got 100 families to send blood samples to them. They made cell lines, and shortly after that, any researcher anywhere in the world that wanted to study our disease could just call up an 800 number, give their credit card, and get the samples FedEx a day or two later. Completely changed the game because I didn't need to worry about people having to suck up to these two researchers. Same goes for other tissue types. Brain tissue is really hard to talk to families about, but has turned out to be really critical for our disease. We can talk about more of these later. Antibodies is an example of a reagent or tool that you need to make. So that in this case, it's something that you use to determine where a protein is located in tissue and whether it's active. And if you're using mice, you need antibodies that kind of act on the missing AT protein in mice. And if you're working with human tissue, humans and other organisms. So this is an example of a kind of one of those boring things, not very sexy, but really, really critical to getting it right. And early on, we had some people make antibodies that weren't very specific. They would detect the AT protein, but they also detected other things. And we had some really bad papers published as a result. So this is an example where you really got to do it. Model organisms. You've heard Matt very proficiently throw out all the Latin names for all the model organisms this morning. I was really impressed. Drosophila. I remember the first time I said that word and said it right, I was proud of myself. Aspergillus, C. elegans, there are all these terms, but basically, yeast, fungus, fruit flies, zebrafish, we've worked with all of them, and each of them has some benefits and some not. Never ask the guy who works with zebrafish if zebrafish is a good model for your disease. Of course it is, it's the only one you need, he'll say. I think what's driven us is we continue to need a better and better model. For a neurological disease, ultimately, you need a model that's very, very close to humans. Our most recent model that was just made last February is an AT pig. They're actually mini pigs, so they only weigh 300 pounds instead of 1,000 pounds. So when they told me mini pigs, I pictured cute guinea pigs, but they weren't. They're still big. So again, the problem we ran into was that even though we've made mice that have AT, the gene is just disrupted and they get cancer and they have immune problems, they don't have any neurological problems that are obvious. And the same thing with the pigs. This is one of the AT pigs. And you'll see that big pink stripe on them. And the reason there's a pink stripe on them is I flew to South Dakota and Iowa to see these pigs because I was told that they had been born. They're all now starting to have neurological problems. You've succeeded, Brad. We've got AT pigs. So I flew there to look at them myself. And I looked and I looked and I looked. And eventually the researcher came over and spray painted pink on all the pigs that had AT so I could tell which ones had AT and which ones don't. So a little tip for you. When they have to spray paint them to tell you which one has AT, it's not a very good model of the disease. Really disappointing. Where we are right now is we're now using that technology that Matt kept mentioning, CRISPR, the gene editing technology that was just invented, to make our marmosets that have AT. We have embryos right now that have the gene disrupted in them, and they're inside surrogate mothers. It can't be parents, it can't be fathers. Surrogate moms, and we're hoping that some AT marmosets will be born in the next few months. They're non-human primates, so maybe they'll have a neurological problem, but this is an ongoing struggle for us. Another example of something that you need to do research on AT, hopefully, is to get to some chemical tools. So in our case, we have some tools, some inhibitor compounds that you can put onto cells, and it'll disrupt the AT protein or inhibit the AT protein, basically cause the disease in that experiment. It's been really useful. A company called Kudos that was bought by AstraZeneca manufactured that compound. The reason they're interested in it is it turns out that AT kids are extra sensitive to radiation. So they're interested in making compounds that inhibit the AT protein that you could somehow deliver to tumors in normal people and make their tumors more sensitive to radiation. It's cool. In our case, I'm not trying to do that. I'm trying to actually restore the AT protein. But having this tool is really useful. And as a little aside story, I'll tell you that some researchers a few years ago took this compound, put them on some cells from some normal people, and then infected them with HIV. And actually, they couldn't infect them with HIV because the AT protein had been blocked. So, You've heard that story with several rare diseases, really interesting. Someone mentioned scientific conferences and workshops, and we have a lot of those. You can have really big ones, especially in the case of our disease. If you let the cancer guys come, they can fill up the room really fast. You can have huge conferences with very structured presentations and poster sessions and abstract books and all that. Or you can have very small focus workshops where you don't let anybody show more than one slide, and they focus on one topic. Maybe a few people are from the AT world, and the rest are from other diseases, but in that particular specialty. For example, last December, we had a meeting that focused on the mitochondria, the things in your cells that produce energy, and how they may be disrupted or dysfunctional in AT. And I brought maybe 5 researchers who were experts on AT to be in the room, but I brought maybe 30 other researchers who are world experts on mitochondrial dysfunction and other diseases, so they could get together and compare notes, and we can make faster progress, generate new research strategies, and so on. There's a whole art, and you could give a whole conference on how to run those meetings. Basically, you want people to be really candid and blunt. I think I'm a little different than some people. Although I always tell my donors and maybe my families about how much effort we put into making people collaborate. Sometimes I don't care about collaboration as much. Sometimes I wouldn't mind if they're a little competitive. If they're really blocking things, if someone who's not going to do anything themselves is hoarding something, then that's intolerable. But there are ways usually to fix it, at least behind closed doors. You can say, here's the deal. If you give your samples and you give your reagents and tools and you do the work, then you'll all be on the paper together, and you can usually figure it out. The harder thing to do is to get people to really think out of the box and not say the stuff they always say at every conference. So if you have immunologists there, they go to immunology conferences every month. And if you have the cerebellum experts there, they go to their conferences. And how can you get people to say, we're in the room together with 2 different backgrounds. What's common about this? Why would one disease have both of these things going wrong? And really get breakthrough ideas. Next thing that I think you absolutely need are shared data sets. And I think there'll be a whole session this afternoon on sharing clinical data, registries, and sharing genomic data. This is absolutely critical. In our case, we wanted to look at all the mutations that have been observed in AT kids. There's some diseases like cystic fibrosis where like 90% of the kids who get that disease have the same mutation, and if you go to 100 mutations, you cover 99% of the patients. In the case of AT, just about every AT kid has a new mutation, so that's been very frustrating for us. It makes it really hard to do carrier testing, but we want to continue to accumulate that data. People, about in the year 2000-ish, there was this new technology called gene expression profiling using DNA chips that became very, very hot. Everybody started doing it on different tissues, disease and healthy, and they did it for AT. So we right away set up a public database where after they looked at their own data and they published whatever they saw, they got to do that first, but after that, then they put their gene expression profiling data all in one place in a standardized way so that others could come along and look at it. Brain imaging data is the same way. It's hard to get one kid who's had an MRI every month for their whole lives. I wouldn't mind it, but it's hard. But if we get a lot of different patients and families to send in their MRIs, we can actually learn from them. The next 8th thing that we need is, and all of us need, are quantitative measurements for the disease. Some kind of care endpoints, or sometimes biomarkers are the right term, but clinical scales, ways to actually measure so that not just us parents looking at our kids, but so that any clinician could tell if a drug is working or not. And it sounds simple, but actually it's tough. In a neurological disease, you can have a really nice description of how you should examine an AT kid. And then if you give that to another neurologist on the other side of the country, and he does the same thing with the same kid, they're going to come up with different results a lot. And it's really, really frustrating. So you need to come up with a way that you can absolutely know that if you run a trial in 2 places, for example, that the results will be measured the same way. Biomarkers are talked about a lot. Sometimes biomarkers are used to tell if a drug is working. Sometimes biomarkers are to tell an early diagnosed person what their disease is how it's likely to unfold. In our case, we just are hopeful for something that can be quantitative that you think is clinically relevant. So you can do a lot of studies and find something that changes with the disease as time passes, but you don't necessarily know if that's a cause or an effect. A lot of things go wrong. And so what you're really looking for is something that you believe is a surrogate for what's actually going on that's causing the disease. In our case, we have neurodegeneration that's going on in the brain, but maybe there's inflammation that's playing a role in that. If that's true, maybe by following inflammatory markers in your blood, you could know whether a drug is working on a kid. This is something that we kind of have, but we'd love to have much, much better. And again, this afternoon, there's going to be a session on it, a database that holds information about HE kids. And because the disease is so rare, as many kids we can find all around the world. So you start with clinical data, how you can get all their medical records. Right now, we're planning to sequence the genomes of every kid we can find, so we want their genomic data. But there are a lot of new technologies coming along. People who are using cell phones to record, to study movement, people with movement disorders, people who are new technologies like Fitbits that you can wear. We want to be able to incorporate all those data. We want to be able to survey our families whenever we want to with new information, new questions we have, put those data in. And then we want to make sure that the database isn't owned by one consortium of researchers who have it like a private club and you have to get in their club to get access to the data. We want to make sure if there's a 14-year-old prodigy mathematics kid in India that has a great way of doing pattern recognition, that we could share the data with him if we wanted to. We've set up clinical centers, one at Johns Hopkins that has a multidisciplinary team of physicians and neurologists, pulmonologists, immunologists, oncologists. And until we did that, typically if a physician had a kid with AT in front of him, that kid was the only AT patient he would see in his career. So now we have a place that's seen hundreds of AT kids, followed them over time, knows what's normal, what's unusual, and has really helped with thinking about how to manage the disease symptoms better. We've done the same thing with St. Jude's in Memphis, Tennessee, where they're one of the preeminent places for managing cancer in kids. So because AT kids have special things like extra sensitivity to radiation, we've tweaked the protocols for most cancers so that if a kid with AT, when a kid with AT gets cancer, there's a team of experts there that can really advise and make sure that the treatment is optimized for AT. Because lung problems are the biggest cause of death, we've also done a lot looking at lung functioning, swallowing. We've also done some studies of cognition because it turns out that even though the kids seem pretty smart, the cerebellum does play a role not only in controlling your movements and stuff, but cerebellum also is now being found to play a role in some cognitive functions like executive function, how you decide between different things, or even some emotions. So we're looking at those things too. And again, you have to return to your mission. So if your mission is just to get to a cure, that's one thing. There could be a mission to prevent the disease. You could screen everybody in America for carriers if you could. Or is your mission to improve the quality of life? One of the things we do frequently is also think about our disease in terms of age groups. So you have the children who aren't born yet. You have the children who are born are still running around, still have brain cells that you may want to keep alive. And you have the children that are already in wheelchairs that it's too late to save those brain cells, but maybe there is ways you can still make life better for them. Brain imaging studies are really important, but not that great. So I don't know if there are a lot of brain imaging people in the room or anybody who's working from a brain imaging company, but you can see me afterwards. Just don't see me when I've been drinking or I'll be really rude to you because the brain imaging study, scientists have the coolest slide presentations and video presentations by far. They're colorful and amazing. But as far as really telling you something at a high enough resolution to know what's going on in the circuitry and what's happening with cell metabolism, it's been disappointing. Again, I mentioned genome sequencing. One of the reasons we're interested in sequencing the genomes of AT kids is that we've found that there's a variability between, even though they all have the same gene screwed up, there's a variability between different kids. Some kids seem much worse, and some kids progress more slowly. And if you look at siblings, they tend to be more similar than AT kids who aren't related. So it makes you think there's a genetic background that plays a role. So maybe if we looked at the whole genome, we may find other places, other loci in the genome that somehow determine how severe or mild your AT will be once that one AT gene is disrupted. Genome sequencing could also be useful, as you heard earlier today, if you do run a trial and you find out that some kids respond and some kids don't, you may find that there's a genetic basis to that. This sounds maybe less scientific and cool, but just improving caregiver understanding of our disease has had a huge impact. We still are astonished by some of the most, and it's not about, you're in the middle of Iowa or something. Every time I say that, someone from Iowa is going to come up to me afterwards. But in the middle of nowhere, and you don't have good care or something like that. No, I can name some of the best teaching hospitals in the country that have misdiagnosis of AT or that have seriously distorted views about how you should take care of a kid with AT. So it's an ongoing effort, and I think you get a lot of bang for the buck. We interact with academia, industry, government, nonprofits, as all of us do, I think. We can talk later if you want on how you go about engaging industry to be more interested in your disease. A big part of our role is to just encourage AT families to continue participating in research. You can give them to give a saliva sample for genome sequencing. It's a little harder for them to give you spinal fluid from a spinal tap, but sometimes it's really important. So now I'm going to tell you just a few examples of what I envision for my disease, what I envision maybe where treatments are coming from. But my main point for you is that even if you're just getting started, and even if your disease the knowledge about it is really, really primitive, you have to have some ideas of what the treatments might be. You can't just be funding research to learn things about the disease, because you can keep learning just stuff about your disease forever. You want to learn stuff that will get you somewhere. And so if anyone stops me, if it's a venture capitalist who has portfolio companies in biotech, or if it's a family member or a donor that wants to know what we're doing, and we only have 30 seconds, like the elevator pitch time, I have to be able to tell them, right now I can see a treatment coming from these 3 areas, but these are the 3 immediate obstacles we're running into right now. Not all the obstacles. We need biomarkers, we need all these things, but just what the one thing that's holding us back right now. If you get really good at knowing that, then you can get a lot of people to start helping you. You can go to conferences like this, and when you show your list of things that you'd like to do and what the obstacles are, someone in the room may say, actually, Brad, you're wrong. That's now possible. Or actually, this thing that this guy's working on may overcome that. So you got to at least be able to envision the treatments. And the one pet peeve I have is I've gone to some retreats for people's nonprofit organizations where their board is there, their board retreat. And at the dinner the night before the board meeting, I'll try to learn something about the disease. So I'll say to the board member I'm sitting next to, so what's happening in the research on your disease? And often that board member will say, well, I can't really talk about it, but go talk to Nancy over there. She's our science person, and she'll tell you everything. I don't think that's tolerable. I think if you really believe in the mission and your cause, even if you're not a scientist, you've got to learn enough to know what's going on, what's there to be excited about, what's to be frustrated about in your disease. So on that note, I have the following 4 paths. So I'm not going to talk about the cancer and lung problems of AT, but just on the neurological part. The first one is to just restore a functioning AT protein. So this is for any of you who have a single gene disorder. This is something that's probably crossed your mind. In our case, how do you restore a function? So the first one is to deliver a working copy of the AT gene to the brain cells of AT kids. It's called gene therapy. People have always tried to do it using viruses usually, where you take a virus that's harmful or make it not harmful, stick a copy of the gene you want to put in there, send the virus in to infect the tissue, and it also dumps your gene in there. So if you put a good copy of your gene in there, it would replace the bad copy and fix things. That's the idea. In the case of AT, what's my obstacle? My obstacle right now is that the best technology out there, the best type of vector or virus that they use, can carry a certain size gene, about 4,000 bases long, and the AT gene is about 9,000 bases long. So right now, very, very frustrating. There are some people with some cool ideas about how you'd break it into 2 or 3 pieces, send 3 viruses in, put them back together again, all kinds of clever ideas. But at least I can tell, if I run into anybody on an airplane who's in gene therapy, I can say, I need to know, do you have a vector that can carry more than 4,000? Oh, you're up to 8,000. Awesome. Call me when you're at 9,000. You got to know what the obstacles are. And a lot of the obstacles, by the way, don't require money. They just require matching your problem with the people who have the answers. Making cells ignore mutations is what Pat Furlong talked about, what they're doing for Duchenne's muscular dystrophy. There are these compounds out there that make all the cells in your body kind of skip over typos in your DNA. So if you have a mutation, they could actually once in a while skip your mutation, and that might cause a little protein to be made, and that would be therapeutic. That's the idea. In our case, what we're doing is we're kind of waiting to see what happens with Duchenne's muscular dystrophy and cystic fibrosis. There's several companies doing that. The moment they get it working, I'm going to be elbowing all of you out of the way to get it working on AT. The last one is to deliver healthy brain cells made from AT kids. This is a very, very hot, trendy thing right now. Most disease organizations have to make induced pluripotent stem cells, iPS cells, from their patients. In this case, you take blood cells or skin cells from a kid, you take them out, you turn them into stem cells in a lab dish, you use CRISPR or something like that to correct the AT gene, and the idea would be to then differentiate or make those cells turn into brain cells, stick them back into the same kid. Because it came from the same original kid, there's no immune response, no rejection, and it could work. It's a great idea. This is really, really early stage, but promising. So that's if you can replace the AT protein. Let's say you can't replace it. So then there's some other ideas for how I could prevent that brain cell death. So the one thing to do is something that's called growth factors. You could deliver growth factors to the brain. Growth factors are in all of our brains anyway. They were discovered like 70 years ago. But the idea would be to put more of them into kids who have brains that are dying. And it's not specific to AT, so everybody working on this could help a lot of other diseases. But we want to keep our finger on this to see if anything comes up that we could do that. We want to try it on our kids. There's a trial being done in UCSF in San Francisco on another pediatric neurological disease where they're using gene therapy that you just heard about, but instead of delivering the disease gene, a good copy of the disease gene, they're delivering a growth factor gene in so that it can be expressed constantly in there. We've found over time that sometimes our AT kids, when they're on steroids for some other reason, like pneumonias or cancers, seem to get a little bit better neurologically while they're on the steroids. So we're doing some clinical trial right now to look at steroids. And on the same, kind of along the same lines, there's a company called Neuraltis Pharmaceuticals in the Bay Area that has a compound that's focused on kind of quieting down your immune response, inflammatory response. And the idea here is that for a lot of neurological diseases, your brain cells die because of that, what different causes, but the neurodegeneration gets much worse because of the way your immune system responds to it, kind of out of control. So that's the idea behind that. And then the third thing is, which is more the typical drug industry approach, is that if you don't have the AT protein and you have this disease, is there any other protein you can change the activity of in cells to prevent those cells from dying? Or is there something abnormal about or a defect in those cells that you can see that if you could just correct it with a compound of some kind, it might be therapeutic? This is the way most drug discovery has been done. And you can use small molecules or something called interfering RNA or antibodies, but somehow to block or change the activation of that protein. In our case, the biggest problem here is we haven't found the protein yet that we think if you inhibited it, it would actually help AT kids. There are a lot of things bizarre and weird and strange and wrong about the cells from our kids, but we don't know which ones are just a result of having the disease and which ones, if you corrected them, it would actually be helpful. So then the next point is just to emphasize that even, the drug industry is kind of turned on by rare disease right now because the FDA can give it fast tracked, and also compared to things like Alzheimer's and Parkinson's that has a lot of mysterious causes, genetic rare diseases should be easier to figure out because we know the starting place at least is this disrupted gene. That's good and all, and I'm thrilled that they're excited about it, but just to be clear, just having your gene doesn't always make things that much easier. Pat Furlong's gene, she said, I think was 1992 they discovered that gene. Cystic fibrosis was that long ago. The Huntington's disease gene was, I think, discovered in the 80s. Our gene was discovered 20 years ago. And in 1995, we found this gene. We called it ATM, ataxia telangiectasia mutated. Just in 2010, I can't show you 2015 because I wouldn't have the room for it, but in 2015, this is what we now know about the disease. So the AT protein plays a huge role in your cells for controlling the biology when you get DNA damage. It senses the damage, coordinates the cell copying divide cycle, slows things down, brings proteins in to fix the DNA. All that goes on. It's a really sophisticated gene role it plays, and it's very famous in cell biology, even though you've never heard of this disease. If you could look on here somewhere, you'll see that ATM interacts with a very famous gene called P53 that's mutated in most cancers. Also interacts and phosphorylates this protein called BRCA1, that's a breast cancer gene. So it's really, really important biology. But for me, I wish it would be a lot more boring. I wish this gene had been super simple. It makes a protein that floats around in your blood. My kids don't have it. We just got to make a milkshake that has it, give it to them, and they'll be fine. And unfortunately, in our case, it doesn't work that way. So, so far, in spite of all that biology that's been done, we don't know how what happens in AT. So we're still trying to figure it out. One other approach we have is to say, okay, we don't know how to replace the gene. We can't keep the cells alive. Those brain cells, particularly what are called Purkinje cells, are going to die in the cerebellum. But maybe there's still something we can do. What happens when those cells die? The circuitry of the brain is screwed up, but maybe there's a way we can correct the circuitry. This would help even the oldest of kids with the disease. And so right now we've been funding some research to figure out what actually is going on, why when you have this gene kill these cells, kids have problems reaching for things and controlling things. How does that actually work? And when I first asked this question with a bunch of molecular biologists and gene people in the room, they really couldn't answer it. They would just give me this dumb answer like, Brad, well, it's killing the cerebellum, and cerebellum affects muscle control. And I go, yeah, I get that, but could you go a little deeper? And what it turned out was I had the wrong people in the room. So then what you have to do is have a meeting where you have a bunch of circuitry type people, electrophysiologists who really know brain circuitry, to start thinking about it. So anyway, that's my sampling of what I've done for my disease and where we're going and what we're struggling with. I can tell you that we've made some progress. When my kids were diagnosed, the median age of death for kids with my disease was 16 and a half, and now it's about 24. We've done this by managing the lung and cancer problems better, but kids neurologically are still in wheelchairs by 9 or 10. They're still struggling with eye movement, swallowing, all those things. Life still sucks. Social lives, as you know, are like almost non-existent after a while. It's extremely exhausting for families. All that continues on, so I'm certainly not slowing down at all. I'll throw out one more thing, and that is we always try to think out of the box. My two sons are still alive, and they urge me to think out of the box. We always see these people on TV announcing new exoskeletons for paraplegics and stuff like that. In our case, our kids don't have a muscle problem, they have a balance problem. So I was at another conference where I saw somebody zip by on a Segway that goes on two wheels balancing because it has a gyroscope. So right now we've got some gyroscope experts who talking to the exoskeleton people to see if you could make an exoskeleton that keeps people up. And we were tempted to call it the Weeble Project. Weebles wobble, but they don't fall down, but I didn't think my sons would like that. Okay, so really back to my story. I'm going to go really fast on this. In the late 90s, besides I kept running my food company, but my night job of pushing AT research, I also started becoming an advocate for a lot of genetic diseases, Capitol Hill and stuff. In this particular case, I was testifying, but all the cameras were on Ben Affleck. And the room was full of nothing but female staffers, go figure. Worked with a lot of different organizations and been on councils and stuff. In 2000, you may remember there was a dot-com bubble, but there was also a genomics bubble. A lot of venture capitalists wanted to get into genetics, and I had a chance to sell my food company. So I got out of that, and I started a company called Perlogen Sciences that did some of the early, very first whole genome association studies with genetics. We raised lots of money. I had many PhDs and MDs working for me. And the cool thing about that was I couldn't really get this company. The investors wouldn't let me work on AT, but I got to hang around scientists now instead of shrimp people, which was a lot more helpful. And I could meet with industry or with drug companies and for business and talk to them about our business deal for appetizers and dinner, but during dessert I could always pick their brains about AT. We worked with lots of nonprofits around the world and companies, and then in 2009 I got a chance to get closer to what I care about by starting a new company with 3 neuroscientists. One of them was a Nobel laureate, and this company was focused on discovering drugs for brain diseases. Sounds a lot closer, right? Again, we couldn't really work on AT, but we worked with a lot of drug companies, did really well, focused on finding drug targets in the brain in different cells, so I got to learn a lot more about neuroscience. And we actually advanced some programs that are now in clinical development. We're now a Parkinson's drug that's in the clinic. The company was bought just 3 years after we started it for a nice return. It was a bummer to me that we advanced the drug for Parkinson's, but I haven't made squat progress on my kid's disease. But the good news is I've now got a lot of neuroscientists in my life. And today I'm now in the process of starting a new company that's is going to focus on rare neurological diseases. And one of those compounds that we have in mind is actually relevant to AT, so getting closer and closer. Again, AT kids keep getting diagnosed and unfortunately keep dying with AT, so I'm not slowing down. And the last thing I'll leave you with on this is that, someone asked, how do you tell donors about how long it's taking you to find your treatment? And I think one way is to show them everything you're working really hard on, and most of the time, they're going to end up, after you do that, they're going to say, well, we're just glad you're doing it, not us. But even if not that, the other thing you should keep in mind is that if you go back and study the major discoveries that have been made in medicine for the drugs that we have on the market today and how they did it, I mean, sometimes they rewrite history, but if you really look at how they did it, most of the time, serendipity and chance played a big role. They thought the drug was going to be good for this, and instead it was great for that. And that's kind of depressing because you'd like to be able to program it all and make it all come out. But on the other hand, it's also encouraging because breakthroughs do happen overnight, and you could still have a treatment. And something big that's being done for Parkinson's could end up helping my disease tomorrow. So discussion, I went longer than I should, but we still have some time for discussions. I'm going to throw up on the screen some topics I thought you might be interested in. You can either ask me questions or we can debate them, okay? So some of them are how I learned the basics of my disease, how you recruit advisors that are actually useful. So you may want some scientific advisors that look really good on your letterhead, but you might want some that are actually smart and have done something recently, didn't do it 20 years ago. What motivates them? How to really avoid gurus. I can't tell you how many people have come to me saying, Brad, our kid was diagnosed with this disease. There's no organization. We just started an organization. We just had our first fundraiser, raised $50,000, and you'll never believe it, but the leading expert on our disease is in our same town, and he's offered to be our advisor, and he's also offered to receive our first grant. Isn't that great? And you go, in what world what are the odds that the world's expert happens to be in your town? Of course, the family also is seduced by the fact that Dr. So-and-so will also be near our kid, Johnny, and it'll be good to have that relationship. But you got to kind of get over the starstruck stuff and be a little tougher. I think I mentioned that I can go on forever about biases, but steering clear of trendy research. Each year it's something, it could be sequence your genome, make iPS cells, or stem cells. Some of this stuff you really want to fund at least one grant for because you have your donors asking about it. They read about it in magazines and stuff. Are you doing anything with CRISPR, Matt? Are you doing anything with stem cells? So you'd kind of like to fund one of those, but don't unless it's really good science. We can have a debate about whether it's good to fund little things or put all your money on one person and let them really be committed, let them have a longer-term commitment so they can hire people and do all that. I think you have to challenge your strategies all the time. I mentioned that you have to find gaps. There are tricks to leveraging your limited dollars so that you can get a whole lab working on your disease even though you only funded one little junior person in them. You can have battles with institutions who want you to pay for overhead and convince them that you don't. You can play tough with researchers when you should be confident about that. I think even the smallest organizations can become bureaucratic. I can't tell you how often I've met with organizations who in June tell me they're so excited because they're now planning their December banquet to celebrate that they're going to raise 15% more money this year. And I say, but what's happened on your mission? Have you made any more progress? And, well, that's like a second to it. And you're going, something is wrong. I'm really a big believer, maybe because I'm a business guy, in number 13, which is treating your donors like investors. And I think it's really important to keep hope and not get burned out. And one more thing I want to show you a slide of is I thought this might be useful to you. So I show this slide to researchers, not to families or donors or anything, but I thought all of you might be interested in. This is my reasons for not funding a research project. Okay? So you worked really hard on your grant, you submitted it to us. It could be poorly designed. They want to study 3 kids and make all these conclusions from it, and statistically it's not any good. Or they're going to work with AT mice to study the neurological problems, and I know the AT mice don't have neurological problems. Or number 2, the project's been done before. Some cases you want to replicate what somebody else did. But if it's like not that same old idea again, you can kick it out. The results, it could be really, really cool science, but number 3, it might not have any next steps. Like, okay, the experiment works, what are you going to do? Publish a paper, but nothing else. So you got to check for that. The investigator could have a poor track record. There's a whole list of ways to do your background checks to find out whether an investigator really does stuff. Sometimes they take the money and don't think they ever have to talk to you again. And then sometimes the budget is unreasonably loaded up, and sometimes the budget's ridiculously ambitious, and they don't seem to care that they know that it's not enough, but they're just trying to put a lot into their grant. But the sixth one is the one that shocks everybody, and that is sometimes your grant is amazing, it's so exciting, and you are spectacular. I can't believe we're going to have you working on our disease. And your institution's famous, and all kinds of famous people are going to help you, and you're going to have all the resources that we're going to leverage. Everything is wonderful, but I still don't want to fund you. And it drives them crazy. But sometimes it's so amazing, it's such a great idea that leaving that guy's office, that investigator is going to do the work anyway. They're going to find a way. They're going to find some money out of their other disease money that they get or something. And when you feel that, you still have to say no. And again, you have to resist the urge, gee, what if you discover something big? I wish I could put it in my newsletter and brag to my donors that I funded it. But I think your donors will be thrilled if you save money where you can. So on that note, I'm not sure how to go backwards, but any questions or anything I said that was outrageously wrong? Or anybody have an idea on how to get a big gene in, or all those obstacles I told you?
So this question just came in from Twitter. So how do you decide between highly funding one of your very promising projects versus many small ones?
So it depends on the project, whether the project is really, really specific to your disease. If there were really one project that was really specific to your disease, you and your very objective, not conflicted advisors agreed that that was the most promising thing and the most critical, I would put most of my eggs in that basket. I would not be worried about that. On the other hand, a lot of scientists will come to you and claim that you need to give them big grants because they need to have long-term commitments to be able to do what they want to do. And a lot of times what it means is they want kind of like tenure with you. And I'd rather have them being a little fearful that if you're not rigorous, we're going to cut you off. So I guess it depends on how much money you have and how important it is, but I would never be worried about putting my eggs in one basket, but there haven't been very many that impressed me that way. I'll tell you one other thing, too. If you are going to put, instead of like 10 grants, put in money into 3 investigators for a longer period of time, probably do it with investigators you've already funded once or twice and you know how things work. The one thing that you really have to separate is there's such a thing as science that you fund that disappoints you because the science didn't work. And you should never blame anybody for that. That's science. But if something doesn't work because the scientists weren't rigorous, they didn't work very hard, they took the money for granted, they were distracted, or they're just not very good at doing science, you have every right to say, cut them off and say, we're sorry, but this is not in line with our mission. And even though I hate offending you or anything else, I feel much better than wasting my donors' money. Any more questions? Yeah.
In hearing all the talks about the medical research and funding medical research, which is obviously of primary importance, I'm wondering, I'm a medical anthropologist, and I'm wondering to what degree are people also focusing on the social science research, looking at people's, for example, coping, obstacles that people face, the challenges, overcoming challenges, raising awareness, which also plays into advocacy. Is there any budget usually for those kinds of projects? Or?
So when I find my billion-dollar whale to donate to us, I'll be able to do I have a long list of other cool things I would do, but I have to say candidly today, we worry a lot about how families cope. How families cope with the problem doesn't seem to be specific to our disease. It's kind of all of us in the room have a lot of things in common. So there are a lot of umbrella organizations and others that can pitch in there so I can save my money for research on my disease. We do have events where families get together, but one thing we've avoided, I hope I'm not going to offend anybody with this, but one thing I've avoided is having conferences for families to come together just to come together, like where they come together and console each other. And they tend to be to me, kind of depressing And so what we do instead is we may have we have a big fundraising event, like in Disney World, we go to their marathon and have a room of 500 or 700 people in the room, who most of them are connected to patients. We're there for the marathon, but of course we still bond, we still support each other, all that. And similarly, we have, in a couple of weeks, we're having a fundraising weekend where we're getting together. And the purpose of the weekend is for families who are planning fundraising events to learn from each other and get better at it. But we have a really good, heavily alcohol-laden evening or two where we talk about our problems. And you have to be really sensitive because parents of 3-year-olds with our disease are really dealing with a different disease than parents of a 16-year-old because of the kids. So. Good. Thank you very much.